Good morning, good afternoon, everybody. Thank you so much to David and Carl for the invitation. I wish I could be there with you. I remember last time I visited you, I think we walked down the street and bought some tarte tatin to have just before the talk. I look forward to repeating that at some point soon. Okay, so I'm going to talk about two stories related to adaptation. First story is about how it's used in the context of sensory detection. So I'm going to give an overview of adaptation and interval correlations, interspike interval correlations. And so the context here is really about how adaptation can enhance signal detection. And there's a puzzle, is that there's approximately independent adaptation states that are part of the sensory cell, give rise to correlated interspike intervals. And this is a result we've known for a while, but more recently we've been able to prove conditions under which we see this, and they're quite general conditions, which I'm going to give you an idea what that's about and the implications for sensory design, sensory system design. So this is a long-standing interest of mine, how one of the reasons I've been working with Len Mailer so much is, here's an opportunity to understand one sensory system in great detail and come up maybe with some kind of standard model, if I can say, for how you mix various synaptic ingredients and plasticities and circuitry to achieve certain goals. So that's been driving me for about 30 years now. The second part is about adaptation, how to compute elapsed time. And this is about timing of encounters with landmarks during navigation. So it's related to path integration and how encounters can produce bursts of spikes just at the gateway to the pallium, which is hippocampal type structure. How you can infer time since the last encounter from the burst magnitude. And you can infer maybe multiple past encounter times to support path integration. All right, now this is an example of an adapting cell. And what we see is, so this is from the mossy cells of the hippocampus. They sit between dentate gyrus and CA3. So what you can see is you turn on a stimulus, step input, and you get a fast firing that slows down. And if you actually measure carefully the thresholds, you'll see that over 8 spikes here, the threshold has gone up by about 9.6 millivolts. And this is known in various parts of the hippocampus. In particular, you can look at different parts of the hilus or Ca1 cells or GC. So as a function of spike number, you see these spike thresholds go up. And in particular, these red ones, the mossy cells, are like the champions of this. They really go up a lot. And we're trying to figure out why that is, what it's used for. You can also plot how that threshold decays back down with time. And the time constants are quite long, about 500 milliseconds. And Mauricio here, postdoc in my lab, has cooked up a model, which will be submitted shortly. And I won't talk about today, but you can, it's the same flavor as some of the things I'll talk about today. But these cells have a few special features, which make them more complicated. But nevertheless, the time constants are quite long. Okay, now, a lot of what I'll be talking about is related to this guy, to this animal. It's a weakly electric fish. So it's a model for studying a whole pile of things, but in particular, it's a model for studying very sensitive detection, even when the firing rate doesn't change. Okay, so the pattern can change, the pattern of spiking. So basically, it's like a visual system or a sense of touch that uses electric fields. When the animal generates this electric organ discharge, which is like a sinusoid from its tail, sets up a dipolar field that oscillates at about 1,000 hertz. When an object comes by, you have a distortion of the field lines, and that produces, for example, an increase in the amplitude of the carrier here. And what is important is that this is detected by the main receptors on the skin, and then The next layer of cells looks only at the modulation. They kind of rip out the carrier. Okay, so it's like you can think of that as a bright spot on a dark background, or you have the other way around as well, more conductive or less conductive than the surrounding water. All right, here's an example of what happens when the animal swims by an object. This will repeat over a few times. This is from Mark Nelson and Malcolm McIver's work. You see the fish will swim backwards to find a prey. And on the body, what you're seeing is a change in potential caused by the prey. And initially, when the animal detects it, it swims backwards to find it. It's in the range even less than 1 microvolt. These are extremely sensitive animals. Okay, and we're trying to figure out, how does that work in particular? All right. So we've built models of electroreceptors a long time ago. What you find is that the interspike intervals set up in bands like this because the spikes don't fire at every cycle of this waveform, but they tend to skip from one to the other. So the intervals will be separated by an integer number of these oscillatory cycles. Now, and what you notice is that if you do a return map of the intervals, you have little islands that are slanted backwards, and also the whole thing is slanted backwards. So there's 2 kinds of negative correlations that are going on. Actually, if you can measure the serial correlation here, you'll see a strong negative serial correlation at lag 1. I'm going to be talking mainly about the big slant, okay, in this talk, the big negative slope here. So you can cook up a model that works very well, reproduces the data. Here's the data, here's the model. You can get things to match up pretty easily. With a bit of work, you need a model like an integrate and fire model, but you need a dynamic threshold. So the threshold gets kicked up at every spike. So you get a spike when the voltage equals the threshold. And this synaptic input here includes noise and also that sinusoidal waveform, right? And the important thing is that the adaptation doesn't reset always to the same value. It perpetuates the memory of the past states. It just adds to where it was. So this is a 2D dynamical system. The voltage resets, but the adaptation doesn't. It keeps going on and carries the memory forward. You can think about these ISI correlations also in sparse networks. That was for a single cell I just showed you, but you can think about sparse networks where, for example, an inhibitory network will set up oscillations. Here's the oscillation in the activity with sparsely connected inhibitory cells. And what you find in these networks is that it's quite regular spiking. This is a distribution of interspike intervals. It's fairly narrow. Okay, look at the times here. But if you look at the correlations between these intervals, there's a whole distribution of them. And sitting around -0.25 is the mean. So in the network, you'll get a whole ensemble of correlation times. The same way people have been looking at distributions of firing rates in networks. And this may be important for downstream targets. So this is work done by my ex-postdoc, Wilhelm Brown. You can find certain parameters that will actually bring that correlation to its most negative value. Okay, and that may have implications for downstream plasticity that's reading out these rhythms. All right, so back to adaptation. So now, if you put a step input, you'll see the firing rate go down. Okay, and there's been lots of work on integrate and fire with adaptation and noise. So these kinds of models where you have a -a here, and a is the adaptation process with its own time constant. So there's been a lot of theoretical work, well, not a lot, some theoretical work on ISI correlations in single neurons and networks, especially by these 2 guys, Thilo and Benji. Also looking at, these are escape time processes in 2 dimensions, they're non-Markovian, and they have interesting evolution of correlations in them. But we're going to put that aside, we're just going to think about the problem of what's going on here. So you have inside the cell some adaptation process that goes up and down. Okay, so whenever there's a spike in blue, you see that the adaptation has been kicked up, and then the adaptation variable goes back down. The whole model fires again because voltage equals threshold. It gets kicked up again, and so on and so forth. And you can see already here where the negative correlation comes from. If you have a short interval here, this is now a high value, will take a long time to decay, producing a longer interval on average. So that's why you get these negative correlations. Now you can therefore calculate the interval correlations here, okay? And this is what's represented here, the interval correlations as a function of lag. And you see that at lag 1, you have a negative correlation. So longs followed by short intervals on average and vice versa. So the question we were looking at is, okay, what does this mean for the actual adaptation states? Are the red dots correlated with one another? The intervals are correlated negatively. What about the red dots? Those are the adaptation states. Are they correlated? All right. So it turns out that they're very weakly correlated. So this puzzle is illustrated here just on the Maurice-Lacar system. You can take an actual more realistic neural system and look at this. So you have the voltage in black, you have the W variable in blue, the recovery, and then you see that if you look at the correlation between the intervals, you're going to get some correlation, but there's very small correlation between the adaptation states. And this is true in the LIF type models as well. Okay, so what? Serial correlations between interspike intervals do not imply serial correlations between adaptation states that give rise to those intervals. So the adaptation states are quasi-independent, and what we've argued is that this enhances the encoding properties of the system, because you're using independent variables to encode baseline activity, stimuli that come by. Okay, we can throw away LIF type models and just deal with non-homogeneous Poisson processes, and that's a lot of what I'll be talking about next. Is you can just write a Poisson rate that depends on an adaptation variable, and when the adaptation is high, then the rate goes down. Okay, so you can use a simple model like that and try and estimate alpha and beta from the data. And you can just therefore disregard the fast sinusoidal driving, and the noise is built in, of course, to this kind of process. And for exponential adaptation, this is what happens upon spiking. The adaptation state gets kicked up to some new value that depends on the interval of time. And there's a relationship between all these variables. Okay. Now, in particular, what we've argued 10 years ago is that if you have a match between postsynaptic and presynaptic timescales, you can recover the quasi-independence. So the presynaptic states in neuron 1 upstream look like this, but if neuron 2 downstream connected to it has the same time constant tau, or some effective time constant tau that looks like it, then these states are going to recapitulate the adaptation states upstream. And therefore you can transmit this quasi-independence down the line. All right, so this is how it would work. You get these adaptation states for the adaptation process. The hazard rate, the firing probability goes up with time, okay? And until you hit a spike, and then it goes down. It's silent for a while and goes back up. And downstream in the postsynaptic cell, you would have, let's say, a proxy sequence of these adaptation states. These states here will recapitulate these states here if you get some matching of the time scale. So this idea would also imply that these states are not very correlated, or they're quasi-independent. In experimental work done back 6 years ago, here with Curtis Marcoux and Will Nesi and Len Mailer and others. We've looked at this in some more detail, and we found that the electroreceptors, they have these negative correlations. They're shown over here. These are the afferents, so they have this negative correlation. But the EPSPs in the next cells show much smaller correlations. They're over here. They're blown up here in the control. Okay, this is fast, 100 milliseconds, this is 10 milliseconds. So in the control, if you look at the successive heights of these EPSPs, they would be like the synaptic proxies here, okay? And these heights actually show little correlation. And we're trying to unpack this whole sensory system because this is very classic. Most sensory systems and lots of transmission in the brain is set up like this. You have AMPA and MDA, GABA, and we poison them one by one and trying to figure out who's important for what. But the main point here is that as you go from the correlated intervals of the electroreceptors, downstream, you get this low correlation between the responses. And that lends support to this design where presynaptic adaptation can be matched to postsynaptic response and thereby enhance detection. Okay, so we wanted to know more recently in a set of papers we're doing to kind of show this in more detail and to show it experimentally. Will, in particular, came up with this theorem, conditions for quasi-independent adaptation states to give rise to correlated ISIs. I'm just going to sketch the idea in a few minutes here. So we're dealing with a Poisson process in which there's a dynamical variable. So the rate is random, is Poisson, but there's a dynamics in it, which is the adaptation process. And between these between spikes, you get this exponential decay. So I'm not writing the differential equation here, I'm just writing the solution between spikes. So you have this adaptation variable, and eta is this adaptation time. And you can think of interspike intervals as differences between successive adaptation times where there's a spike. So you have a stochastic spiking rate that depends on this adaptation. So for example, you can have this classic exponential form. And from this, you can derive the survival function. So like the system goes on without spiking, and you get this expression, which is conditioned on the initial time. And it has this potential here, which is the integral of the rate, okay? Now, this adaptation time is kind of one of the novelties we think of this approach, is that we're moving time from absolute time to a time in the reference frame of the adaptation. Okay, so this is a very busy plot. I'll just try and give you an idea of what's going on here. So on the y-axis is the adaptation time course. So you see the big black line. This is, let's say, how the adaptation decays after a spike. But it's the workhorse of the whole process. Okay, so it's this curve. And this is time if you want, but we're writing it as adaptation time. And there's two sets of squares here. There's like the red ones and the blue ones. So let's start with the red ones for the first spike. So here at this point, the system spiked. So what happens when this system spikes is that the adaptation variable gets reset through this function g to a new variable up here, to a new value up here. And to this new value corresponds an internal adaptation time, which is back over here. So eta 0 star, where the spike occurred, gets mapped through the function f to this new time associated with the new adaptation states. So there are two maps operating, the one for the adaptation states and the one for the times, and they're related, of course. And then the system will decay back down, and at some point it'll fire again over here. And then the same process repeats. The adaptation state will get updated up to here, and that corresponds to a new time. And what you can see here on the bottom plot is the survivor function as well as the spike rate. So as the adaptation decreases, the spike rate increases. is what you expect, right? But the survivor probability goes down. Okay, so that means there's less and less chance of not having spiked. And this is the area under the curve, which kind of underlies the theorem, where we say, suppose that for a given tolerance epsilon, there exists a particular eta such that this thing, this potential is less than epsilon. And so I won't go over the details here. And you can suppose that this map f for the times is less than this value. Then the survivor function can be expressed in two pieces. So basically we're saying there's a piece here and there's a piece here on both sides of this thing. And this is the amount that we're going to be willing to neglect. So basically, we can show that the survival function is epsilon close to unity. That is, this thing over here is less than epsilon. If this is less than epsilon, that's good, because that means that P of eta given eta 0 is about P of eta. So you get rid of the conditional and you have independence. But it's going to be quasi-dependent. There's a little epsilon error in the picture. So basically you can show that P of eta minus this conditional is going to be less than epsilon. So I don't have time to go into the details. It's a fairly long paper, but that's the idea based on this kind of construction. And so this implies that for sufficiently small epsilon, you can replace this by that. And this means that the adaptation times, and by construction, the adaptation states, are quasi-independent. You can look more into the technical details. If you're interested in this, you're dealing with Markov transition operators for the times in that map and for the values of the adaptation states in that map. So there are conditions where you can go from this description to one without the conditional, and same for here. And you can write these operators, for example, in this form using all the stuff I just showed you. There's this Rouel-Perron-Frobenius operator theory that guarantees the existence of invariant asymptotic densities for these operators. And these operators are going to be fixed point of the Chapman-Kolmogorov equation that governs how these times and H values, how these two maps behave in time. Okay. So that's more technical. And what you can, a corollary of this is that if a point process satisfies this property, this independent, identical, distributed variable property, then the sequence is serially correlated, meaning that subsequent ISIs will be statistically dependent. However, non-subsequent ISIs are quasi-IID and therefore uncorrelated. And you can show that these correlations of the intervals are going to be between -0.5 and 0. With a lot more calculations, you can also show that the Fisher information associated with this patterning due to these adaptation states is higher than if you had just a renewal theory, and higher than this other one, this other rate, which has variance reduction because of the negative correlation, but not quite the whole picture. So I won't go into the detail here, but basically you buy about 20–30% more information with this pattern code. And we're particularly interested, and this is going to be forthcoming this year, is what happens when the fish tries to detect, let's say, a 5 microvolt stimulus or a 1 microvolt stimulus, and you're looking at the changes in the firing rate. And at some point, you just can't see them anymore. But it turns out that the pattern code is going to be able to pick out this limit of absolute detectability which is around 1 microvolt. So that's where we're heading with this. Okay, now let's just do a little sidestep. In parallel to all this and related to all this, but we're not completely clear how, is that there's also noise in the postsynaptic target that may be helping the detection. This is an old idea. I've been working on that on and off for about 30 years. It's called stochastic resonance. But what we've noticed in the postsynaptic cell, now this is in the pyramidal cell downstream, is that there's a varying amount of noise depending on where you hold the potential. So for example, at minus 20 from threshold, you get, this is like minus 70 or so in absolute millivolts. You see some fluctuations. And as you get closer and closer to the threshold, you start to pick up a lot more noise. And you start picking up these little spikelets, which we call blips. We're not clear where they come from. They might be dendritic spikes. It's not clear. It's still work in progress. But there's this noise variance and these blips that are coming up that we think are helping for the detection process of these very, very small stimuli around the microvolt range. And we developed a method to figure out, well, what exactly might be going on in such a system. As you're getting closer to the threshold, are the sizes of these blips going up? Is the noise really going up? Or is the drift in the LIF model going up? So we looked at the data a bit more, and we see that as we go closer and closer to the threshold, the variance is going up and up. So this is an example of the membrane fluctuation distribution when you're closer to when you're far from the threshold. And as you get closer to the threshold, you see that it gets skewed and you start picking up more and more noise. And look at the variance has increased quite a bit here compared to there, okay? So we've devised a method to figure out what has changed as you get closer and closer to the threshold. And so there are these blips that happen, and we're trying to figure out, well, what's a blip and what's not a blip in here. So what you can do is do the standard thing from stochastic processes, is look at the difference process, so the increment process, and then set a threshold. And you can analyze this set of jump distributions. So like, the distribution of the size of the jumps and the times of the jumps. And they seem to be randomly occurring, these jumps. Are they helping the detection process from signals upstream? We don't know, but this is our best guess. So the jumps are detected by applying a threshold on the increments, and this creates false positives too in the problem, because you're going to get false positives just by the diffusion process here. So we developed a way to tease out the false positives, and this is work by my ex-PhD student Alex Manasson, where you can estimate the drift, the noise, the rate of jumps, and the distribution of jump amplitudes using a jump diffusion equation. So there's a diffusive part, it says LIF part if you want, diffusive part, except we allow for nonlinearities here. And then there's these jumps in there. And I won't go through the details, but we developed an iterated Fokker-Plonk analysis to remove these false positives as best we could, to come to the following conclusion. As you go closer and closer to threshold, the jump rate goes up. The jump amplitude doesn't change much, but the noise intensity goes up. So as the threshold is approached, the drift function stays the same, roughly. The diffusive noise increases and the jump rate increases, but the size of the jumps is about the same. So this is consistent with a picture where the system is somehow pouring in more noise near the threshold to be able to detect things better, perhaps. So this would be what we call noise, voltage dependent noise to detect the single peri-threshold. So stochastic resonance, peripheral to superthreshold stochastic resonance. So the picture that emerges is that you have primary receptors that appear to be superthreshold, because the models I showed you are actually suprathreshold. And that's because they deal with the carrier, this relatively large carrier generated by the fish. So that's a suprathreshold signal. But the second order cell, it appears to operate more perithreshold. And it deals with the modulations, including the very small modulations caused by stimuli. So this is maybe an arrangement where you have suprathreshold dynamics at the periphery, and then the next station you have subthreshold or perithreshold dynamics, where noise can be assisting the detection process here, and the noise will depend on the mean voltage. Is this a general principle? Consequences for detection and coding, how it interacts with our other story about the interspike interval, correlations, and enhanced Fisher info. That's something we're still working out. Okay, so that's part one. 30 minutes here. I'm going to go on for 15 minutes for part two. Now, and while I'm at it, is anybody still there? Can people hear me?
Yeah.
All righty, thank you. Okay, so this is like teaching online these days, right? You have no idea if anybody's out there. Okay, so the second part is about adaptation to compute elapsed time. So the context, this is the slide repeated from earlier. We have a timing of encounters with landmarks during navigation. And the animal is path integrating, trying to figure out where it is to head to some final goal, where there's maybe food. And I'm going to talk about work where we focus on a structure that bursts. It produces bursts of spikes whenever there's an encounter with a new object in the environment. And these bursts can be used to infer time intervals since the last encounter. And more recent work, this is published work, and more recent work is pushing this in a direction where maybe these cells can provide a representation of sequences of time intervals. There's been recent work, very recent work by Belek and other Wolfgang Mass's group along the same lines, but we're not doing a recurrent neural net at this point. We're talking about what this system is doing, how it's packaging, representing the intervals using adaptation. And it's going to be very long time scale adaptation, as you'll see. Okay, so in particular, what we're talking is going from time to space. So we have timestamps for spatial encounters. So I meet this landmark, I stamp it. I meet another one, 10 seconds later, I stamp it. And so the adaptation will be representing the time between the encounters. And if you think about it, when you're navigating, there are landmarks that help you navigate. But if those landmarks show up at odd times you've not used to, then there's something amiss. So somehow the time stamping is part of your internal representation of where things are. So this is what I'm talking about, is that time representation. And there's been lots of work by Moser's group and Hamerl and others, not lots, but some work on the time representation, which this is part of. So this work is done mainly by Avner Wallach, who's a postdoc at Columbia now with Nate Sautel, and also by my ex-PhD postdoc with Len and I, and James Jun, who was a PhD student in physics with me and Len. So you may, if you've heard about the neuropixels technique, you may recognize the name. That's what he went on to do after his PhD here over at Janelia. Eric is our mastermind molecular biologist, electrophysiologist, research associate, and Raphael is a PhD student in my group. And this is Len, who I've been working with for over 22 years now. Okay, now here's a fish. Here's an arena. It's about 3 meters wide. It sits here in the Faculty of Medicine in Ottawa. And what we have are little landmarks. And there's food that was right here. the animal learned that there was food right here. So it leaves this home base. There are 4 different home bases, but let's say we chose this one, and it's going to leave the home base and go and find its food. Okay. And as you will see, the fish will be color coded as it's swimming. And it's color coded in terms of the pulse rate at which it samples its environment. This is a slightly different electric fish. This is from the what are called pulse type electric fish. And when they're interested in something, they increase their pulse rate. It's like they're strobing the light faster to sample things. So as they're moving around, they're changing their strobe rate, their pulse rate, and let's see how this goes. So the strobe rate is going down here, 75 hertz, 77. It's moving around as the fish swims. So look at what's happening. You'll hear it maybe a little bit at the beginning. And here we go. So what happened here is that the food used to be here and it's no longer here. That's what we call a probe trial. And the fish is really not happy about this and increases its sampling rate at this point. It doesn't seem terribly hungry because it's kind of wandering around before finding the food, but it may be a bit lost beginning and it's just going and finds its bearings with these landmarks and finally gets here. I'll play that again for you. All right. Okay, so that was the PhD work of James Judd in particular. So he helps to have an undergraduate degree in physics and in computer science, and then go on and do physics for a PhD. To do this kind of work was really nice tour de force. And so what we're doing is trying to figure out basically allocentric representations, so relative distances between objects. And the distance traveled and the time between encounters are of interest here. So there's a speedometer, which is the pressure detectors on the side of the fish on its lateral line. And as you saw, the speed is relatively constant, but it does vary a bit. And the timer is what we're proposing is the adaptation. Now, the way this system works is that you have the electroreceptors on the skin, electroreceptor afferents, come into these pyramidal cells, which is what I've shown you a few minutes ago. These are the ones that are using noise and blips to detect the signals from the afferent periphery. They look only at the modulations. The carrier is now long gone. But now we're going on further to tectum, optic temptum, torus, which is inferior colliculus in mammals. And it all comes down to this structure here called PG, which is the gateway to the pallium. So from the midbrain, you go through this bottleneck into the telencephalon, okay? And these are the cells that are crucially important. They display mixed selectivity. It's a paper that's going to come out soon. They also do this. So I'll show you in a second. So we're going to assume neurons can compute the elapsed time between the last encounter, object 1, and a new encounter, object 2. So then the relative distance between object 1 and object 2 is the travel speed times the elapsed time, right? So there's 2 kinds of cells. There's some cells on the fish where we're bringing the object close to the animal, and then we're moving it away. We're representing that on the other side of the animal, because obviously we don't go through the animal, but the stimulus comes in and then it moves back out. And you'll see the firing rate go up for some class of cells that are inside the receptive field, but other cells outside the receptive field don't care. So these are topographic cells. Most of the cells are not topographic. You put at the head or the back of the fish or anywhere, you always get the response. So in a way, space has been stripped away and you're left only with time. It's going to be this time where something happened. I encountered an object. Okay, so the experiment proceeds like this. You do a distance going down with time. So you're close by, and then you move away, and then there's some other random interval, and then you bring the stimulus in again, and then other random interval, bring the stimulus in. And whenever you bring the stimulus in, there's a pile of spikes. Here there's a pile of spikes. That's what we call the bursts of spikes, okay? So the neural responses, if you plot them as a function of the immediately preceding time interval, you'll get a curve that looks like this. So thereby you can do, presumably, the decoding with this. If you know the bursts of spikes, the number of spikes that bursted, you can read off from this plot and say, okay, this was 6 seconds between the interval, since the last burst. Okay, and how can this happen? Well, there has to be some memory in the system, obviously, and the adaptation is this memory. And the adaptation, because you see, if you just leave 1 second, then the response is very small. But if you leave 30 seconds, then the response is quite big. The system has recovered. So there's this representation where you can map time over to number of bursts, but look at the time scales here. This is like 30 seconds. We could have gone longer, but we didn't. So then these are adaptation processes on time scales at tens of seconds. Okay, not 500 milliseconds like in the hilus, or much shorter that we see in many other systems. And if you look at the correlation between these two things, it's going to be good only for the immediately preceding time interval, but not good at all for all the previous ones. That's because we selected certain cells that seem to be blind to the past. They only care about the most recent interval. So the model that we came up to figure out how this might be working is a maximum likelihood estimation model. So it's a Bayesian analysis where we have a latent dynamic variable, which is governed by this. This is basically a modification of Sodek's Markram 1977 paper for 1997 paper, where X tries to go to 1. Whenever there's a spike, it goes down. Whenever there's an encounter, it goes down, and so on. And there's this beta here, which is the memory in the system. If beta is 0, you care only about the last interval in this latent representation. But if beta is not 0, then you care about the previous ones, and then the system has an infinite memory. This X turns into a firing rate with a thresholded rate like this. And then this rate is going to be the parameter of a Poisson count distribution. Okay, so that's a very simple model, simplest, and it's quite noisy because this is a very noisy distribution. So we're making our life pretty hard. And with this, you can do the analysis, find the maximum likelihood for the problem, and you can find that, you can map the real time with the maximum likelihood estimate time fairly well. And furthermore, you can look at the temporal error as a function of time. Well, that error grows with time, like all estimation processes in biology. But the analysis reveals that 500 cells are going to suffice to account for the error that we see experimentally when the fish is swimming around, and what's the error it's making on where things are. And there's about 9,000 cells available, so this is more than enough. Now, the next question, so the way we did this is we just write the likelihood of given these times. So likelihood of these times given the responses is the probability of the responses given the times. Just flip around Bayes' rule. And here you just write down the Poisson distribution in which you've stuck in these other variables. Lambda and X is given by this, but you're using only this part here. There's no beta. And with this, you can find the maximum numerically and for each interval, and you can do some theory and show that this is a pretty good estimate of what's going on. And that's what was plotted in these things, in these parts over here. Okay. Now, the cells that don't have memory are only a fraction of the cells. A lot of cells do have memory. So the last thing I'll show you in 2 minutes is what happens when you include these other cells that do have memory. So then in the likelihood function, you're representing this activity, this rate parameter for the Poisson density by something now that has more depth to it. It has the dynamics, has the adaptation dynamics stuck in here. And beta is not 0, or some of them have 0 and some of them have not 0, so on. So this is where we're at. So this is work that Raphael Lafont-Mercier is doing for his PhD. So the model is that given the response of the cells to the nth stimulus, the likelihood of having some sequence of time intervals is given by this, which is this probability, which is this. This is what we're doing. And these are the responses, the spike counts. And then the estimates are found by maximizing the likelihood. And a simple model, and I'm just giving you a taste of what's to come, is a simple model can actually get estimates of at least 2 intervals from a single stimulus. So you can look only at the most recent response, and in that response lies information about not only the last interval, but the previous interval. And this is what is shown here, where you have, let's say, if you use a single population of cells, and they're all identical. So what we're plotting here is T1 versus T2. There's only 2 intervals. So you present T1 and T2. So you get 2 responses, but you look only at the last response. So if there's no memory in the system, then your last response is here, but you have no idea what the previous interval was, right? T1 can be anything because there's no memory. You can include some memory, and now you're going to start bending things over. So now you have a bit of you lift the degeneracy. You can start seeing perhaps some map, but the color is all blue pretty much. And it's hard to pick a maximum, okay, to find out what was the previous interval. However, if you now ascribe one population to be memoryless and another population to have memory, then you start getting these little bands. So now there's a maximum of the likelihood. We blow it up here. So if you have 2 populations, one without memory, one with memory, then you have a likelihood that's peaked around here. And this is a specific point now that says T2 equals this, T1 equals that. And as a proof, here's an example of time interval estimations for a simple model of 10,000 cells. Half of them are memoryless. The other half have this beta parameter, the adaptation parameter of 0.39. It does very well remembering what was the last, or representing what was the very last interval between encounters, and it can still get pretty much the previous one as well. Okay, so this is therefore another possible use for adaptation, this time on very long time scales, that can help provide support to path integration and path learning. So calibrating with timestamps where I am. Which just brings us back to our current direction, which is really about mossy cells, and well, it's also about mossy cells in hippocampus, hilus, in mice actually, and where you have this adaptation process on timescales of half a second, and we're trying to figure out how this helps now in path integration with behavioral experiments as well as experimental electrophysiology to find out what is the representation going on in this part of the hippocampus. So this conclusion for the last part is that adaptation acting on long timescales can represent sequence of intervals between encounters, and this can support path learning and path integration. And with that, I thank you.